Alright, so it's basically like a test. If that video was like, this is just a test. Yeah, so I've been studying for MCAT. I mean, I told myself I would start. Oh yeah, by the way, like if you have no idea what's going on, so yeah, man is gonna be taking the MCAT in like I think like 6 months. So I'm planning to take it like be ready by it by August because I'm applying in 2027. So I'm trying to get it done by like September, October ish. So I want to be ready by like August. So yeah, let's just make the whole schedule and everything. And I'm just keeping myself accountable. And so this is why I'm having this whole like webcam shit going on. Yeah, so like I started studying like 2 months ago, but bro, I barely made any progress. I wasn't consistent at all. So this is why I'm just doing this. Alright. I have all the UWorld books, so just gotta get through them, basically. Alright, so today is right, March. I'm gonna try to follow the schedule like to the dot. Today is March 3rd. Be ready by August. I don't know, fucking August 30. That means I give me April, May, July. So that's like, this is 6 months right here. I gotta get through biology, chemistry, biochem, and then physics. There you go. Basically, I just have to do it. Like it's kind of hard, but it's really not that hard. Read on the side, like read every day. Plus Anki for psych. I already did the Anki psych deck, but you know I wasn't really consistent with it. No, actually I finished it, but I haven't reviewed it in like a long time. Actually, that's what happened. My bad, sir. Yeah. Okay, so I'm on this page. Yeah, I'm like right here. I'm on page 352. Damn. So I have to I gotta set like goals and shit. Like so I don't actually like. I don't want to just have no goals. I want to like push myself and try to get through a lot. Biology. Right now, page 352. So there's what? So there's 600 pages in this one. 633. So it's almost. Alright, so if we somehow can do 30 pages a day, I think that's not a bad goal. 30 pages a day. So 30 pages a day is gonna be, what is that? That's gonna be around like 30 pages a day. 352 to 630. 630. That means March 3rd. March 3rd. Even 20 pages a day, bro. Like, genuinely speaking, 20 minutes a day, 20 pages a day. Let's just do the math, right? Alright. 630 minus 350. 350. So, if I do 20 pages a day, hell no. There's no way I can finish in 14 days. There's no way I can finish in 2 weeks. That's crazy. That's crazy if you're gonna think. It's not a bad goal though, and I feel like that's achievable. That's not like in the air. Yeah, bro. Like honestly, I think I could do it. Honestly, I believe I could do it. Honestly, 20 pages a day. Okay, so that means right. March 3rd. 20 minutes, 20 pages a day. That means where was I? I was here. 250. Okay. Yeah. So that means I would. I think that should be done by I think with genuine focus, this can actually be done. Not just me just rambling, not rambling, but like just not me just like, you know what I'm saying, just like half-assing it. If I genuinely sat down for even 15 days, even 20 days is not a bad goal. But I'm not gonna lie, with this shit, bro, you can't just keep learning, but you gotta move on. It's 20 pages. Like, I gotta think like about myself. Like, could I really learn this shit? Yeah, but honestly, I could do 20 pages. Okay. So that means 14 days. That means by March 17th. Okay. Done. Like, I swear to God, if I'm just focused and just get, I can finish it. March 17th. I'm already like halfway through it. It's not like I didn't do shit. Done by March 17th. Okay, then we move on to chemistry. Chemistry. Failed chemistry. Passed mathematics. Money count. Do that. Add it. I bought a carpet from Aladdin, so I can finesse and do magic. How many pages is this one? This one's 359. Ooh. It's low-key long though, right? I honestly, I low-key learned this already. But you acting like you didn't learn this, like, regardless, I think 20 pages a day is not a bad goal. Like, it's a pretty realistic goal. I think it's not like, I think that can be done in 4 hours of just like learning and moving the fuck on. So, yeah, so this is 360 pages, I think. Oh, which one is this one? Yeah, 360 pages. 360 pages. Let's get that blue light shit. What was it called? Like flux, right? Where is it? Ooh. Well, if you have 20 pages, that is gonna be like 18 days. That's gonna be April 4th. Wallahi, bro, like if I lock in, I can lose. I'm not even trying to like biochem. This one's pretty long. I ain't gonna lie. $587. 600 pages basically. 600 pages. It's gonna be 30 days. Done by May 4th. Okay. So if you, bro, I'm telling you right now, bro, if you understand all this shit, bro, like that's guaranteed like 520. Bro, this is like this is literally the entire exam. And then so on the side, I'm obviously gonna be reading. So I have all these books. I mean, I've been like trying to get into reading. So I'm also gonna read on the stream and just keep reading shit. Bro, Loki, I'm a little bit excited. I'm not gonna lie. Because I'm not, my whole past like 2 months ago, I just been feeling shitty because I know I've been like, I made this entire, I think I did this, I made like a doc before. I made a doc before to like, with all like the yearly goals, 6-month goal, and that shit fell off, so. But now like, I'm not saying something different, but I'm gonna do it. I don't have a choice. I don't have a choice anymore. Okay. Yeah. So if I'm done by then, what else is there to do, right? I'm gonna have reading done on the side, obviously. So okay. And physics. Well, physics is kind of confusing because, well, you have to learn it. I'm planning to do all the practice ones. Textbooks, so I don't know when I'm done with that. Realistic goal, if I were to lock in and just do physics my whole like day, realistic goal I can be done by June. I I could be done by June. Maybe 15th. June 15th. I could be done with it. I could just learn all of physics. If physics lock the hell in. Just do physics. Realistic, bro. Realistic. And post solution on YouTube. Okay. Well, by then, right? So if I'm done by June 15th, and I want to be ready for the MCAT by August, okay? That gives me, what are all the MCAT dates? Sorry, that's an important question. Man, these assholes don't even schedule. My bad, I didn't mean that. But they don't have it in October. You only can take it in September. So there's no like, I want to be ready by August, bro. You have to be ready, bro. Like, that's the only day. Fuck, bro. That's it. That's like all you got. And then you gotta wait until like January again to take it. Wow. So I really have to take it. I gotta schedule it. So I actually gotta schedule this exam. Okay. So what? The deadline. I mean, you can sign up like us. Okay. Yeah, man. That's why we're doing this, man. It's all or nothing, basically. It's all or nothing. So that gives me what, like, another. How much time do you world? That gives me July. Bro, that's dead ass. Like another 2.5 months. I mean, if you take on the 12th, well, that gives you, so worst comes to worst, we take the exam on September 2nd. September 12th. September 12th. One last minute, bro. Yeah, I'm bugging. I don't know. I'm even thinking about this shit. Yeah, August 12th exam day. So that means, what the hell does that mean? Okay, September 12th exam day. Today's March 3rd. It's basically like 190 days. Imagine having 190 days of live stream. It's pretty cool, actually. Alright. Well, that gives me from June 15th all the way to September 12th to max out. So yeah, I am planning to do like EMT school. That is gonna be something. And that is 6 weeks as well. So honestly, fuck it, bro. Who cares? Since I mean the EMT school is like 6 weeks, I don't even think the material is that hard. And by that time, I think I should be done with memorizing material. And I'm just practicing as much as I can. So I don't even think I think I already can handle all that information. Regardless, max out UWorld plus practice plus reading, just reading plus I don't know, meditation, all that shit, bro. Just do it all, bro. Honestly. Okay. September 12th, max UWorld plus practice, reading, meditation. Bro, by then I should be like meditating like 1 hour a day, bro. Some Naruto. What was that arc when he went to the frogs? Alright, what's next, right? Yeah, so, I mean, now the only thing to do, right, you really just gotta start. Okay. So, September 12th exam day. September 12th exam day. means what? Alright, I think we can start. I think we could just start. That's one of Hold on, take off. Hold on, take off. On it. Yeah, bend over. Jump to the corner. Hocus pocus, pull up in the lotus, my diamond. Hold on, take off. Hold on, take off. Hit it. Not running off. It is on. I'm long. Fast, bend over. Jump with them. Alright, so basically we have to get to 20 pages a day. Chain regulation development. So you go for fertilization. Fertilization is like, so I think it's methylated because bro, there's no way I can't focus with somebody, bro. Just like speak out loud. What exactly? What's the point of this, bro? Those are the reasons. I remember that had a good reason, bro. This section answers one question. How do identical cells with identical DNA become different cell types? Okay, by controlling which genes. Okay, so yeah, how do all the identical cells with all the identical DNA become different cells? Okay, by controlling which genes are expressed with another which from trying are delivered not used on DNA. I got limited lives on genome really. So yeah, first the embryo. Okay. This whole point is, so we all have the same exact fucking cells, and they all have the same DNA. So how do we get different cell types? So first, when the embryo is born after fertilization, well, I'm pretty sure all the chromosomes are super dense, so it doesn't transfuse the DNA, so it uses all the mRNA from the mom and proteins, and around the 4 to 8 cell stage. That's when you have embryonic genome activation. Okay, so that's one. So 48 cell stages when the genome turns on. Before embryo can describe, well, before the embryo can actually do shit with that, they can do transcription. Well, the chromatin has to become loose, and the DNA needs to be accessible. So this is basically this epigenetics. So the epigenetics here is DNA methylation. Gene silencing at Cat So basically, methylation turns DNA off, remodeling. Okay, most DNA methylated. So after fertilization, most DNA is demethylated. Well, why is it demethylated? Well, because your parents they also had their genes are all like that has epigenetic shit. So so we want to just demethylate everything, just make it back to normal, right? I'm confused though. Like, we're talking about methylation and then talking about demethylation. Okay, these are my questions, right? I'm confused. You talking about, first of all, I'm confused. First of all, what is methylation? Explain super concisely. Why does putting a methyl group make it more or less accessible? Explain. So it makes sense concisely so I can never forget. Number 2, why is it cytosine methylation? What does that mean? Number 3, you are talking about demethylation here and methylation here. What's going on? Alright, let's see what this guy has to say. Okay, DNA methylation is adding basically a methyl group to a cytosine D base in DNA. Cytosine CPG cytogenexes DNA sequence is a chemical tag of cytosine because cells have specific enzyme DNA methyltransferase. Okay, why did I ask? It does not block. It doesn't. Certain proteins specifically bind methylated DNA. Bro. After fertilization, most DNA is demethylated. Okay, I have no idea what the hell is just said right there. Adding some cytosine base. Okay, what does that mean though? Or is that good or bad? Does that make it? Okay, if I can't understand this, I'm just retarded. This chemical tag added. Because cells have a specific enzyme. This 5-carbon cytosine. Alright, so it's fine, alright. We have the cytosine. And we just, you know, just do our thing. What does that do though? Okay, regardless, we gotta decide it. Does not block it. It changes how proteins interact with DNA. Well, to retract fresh proteins. Well, certain proteins specifically bind methylated DNA. These proteins occur in stone cases, latest compartments. So it makes chromatin tighter. That's the main thing. Also, if methylation makes it more tighter, then demethylation makes it more accessible. Right? That should make more sense. Yeah, it's gonna activate genes. Okay, look, let's go back to the jump. Most DNA is demethylated. Resets. Okay, reset. Okay, after validation. All the DNA is demethylated. Program. Some genes keep methylation. Yeah, Allah. So the thing at the beginning, everything was demethylated, so to reset it. Alright. Some genes keep methylation. Some printing, later, selective incision, pipe. Okay. Be died and choose cell. Early embryo does not use its own genome immediately. Early cleavage, G code, rapid diffusion. Okay. Why the hell did this happen? Very cool shots. I think sometimes this is a low-key G topic. Yeah, I mean, number 45. So right here, right, basically, yeah, my parent, if dad's comes up, I'm pretty sure it's the exact same RNA transcript. Well, since mom is methylated, she's not gonna have. That's just gonna be there's no gonna be mRNA. Well, so this is paternally imprinted. I think to remember here is maternally imprinted means you're not gonna get shit from mom, and paternally imprinted means you're not getting shit from dad. Because of the differences in primary sex determination, individuals with two. Oh, this is ChatGPT down. Okay. I should answer. I mean, this is the main thing, right? Yeah, that's basically what this is saying. This is motherfucker. There's no point. And we could throw like a picture outside the scene, right? What was that? I guess this is exactly what it's saying. At first stage. Okay. Yeah, that's basically what it's saying. I don't want to delve deep into this shit, bro. There's no point in writing all this shit down, bro. It's just a waste of time. I gotta watch my. I think I. Yeah, I don't care. Okay, so at first, right? What's the. I'll keep it right. طيب شو يعني alright let me read it first because of the differences in primary sex determination why the fuck are you XX individuals? Isn't XX individuals girls? Well, XX cells. Okay, what? I have two X chromosomes. XY, I have one two chromosomes. Without correction, XX cells would make double the amount of X-linked gene products. That would break normal development. So the embryo must equalize XT dosage. What the fuck does this mean, bro? So maybe this picture will help. How the... والله تعبان كثير تعبان. شايفين الوقت؟ هلا الساعة 4 الساعة 4 إلا 5 دقايق الصبح. فطور مو فطور، سحور في ساعة. اوكي في هنا 2 كروموزومز، عنده وحدة اكس كروموزوم، فكل الاكس كروموزوم عنده 100 ايش اكس كروموزوم. اوكي Alright, so basically, whenever you make proteins from the X chromosome, you're just gonna get like double of them. So that's why like females feel like the silence will have to silence them. I kind of get it. This is the main thing. If an XX individual is heterozygous, I don't know heterozygous. I think heterozygous means like whatever heterozygous means, one of the chromosomes is a mutant and the other is normal. Okay, so if XX individual is heterozygous with an X-linked trait, that means they have a 50% chance of having that. The fuck are calico cats? Sorry. pencil cats. Oh, they're cute. لسه هاي كتير حلوة. شايف لونها؟ اوه رهيب. طيب كاليكو كاتس. Roots. Be concise. Concise. Put it all together for a clean screenshot. Roots of Mosaic is tell me why we don't want double amounts of protein. How come? Is it? What is it? Fucking 48, 24, 23, 46, 46 chromosomes. 23 and 23. So, if you are a girl, right? How the hell does that work? You're a girl, you have X and S. Why can't you just have protein made from both of them? 'Cause, like, what the hell? I don't get it. Every somatic cell has 46 chromosomes. Well, 23 are from mom and 23 from dad. These arrange as 22 pairs of autosomes. So, yo, I'm very tired right now. I'm not gonna lie. gonna just chalk this shit right. That's 30 minutes. How the fuck is 22 chromosomes? What? What is going on, bro? What's going on? What the fuck is an autosome, bro? Get this shit, bro. I don't even understand this sentence. I don't understand this. Get this, bruh. How do you go from 23 to out of that 22 chromosomes that get from each parent, 22 are regular body chromosomes. One is a sex chromosome. So each parent gets 22 autosomes, one sex chromosome. to back to the topic, what the kind of lost the plot. So now, how is it double? Okay, now that. Yeah, I understand. I understand it now. Is it so that females are treated like males, so like all that shit? Alright, let's move the fuck on, bro. Like, I'm just wasting my time. This conversation. This is a waste of conversation. It's a waste of time. you read this? Alright. Regulatory elements also contain hence sequences bound by such as level D and sequences. All I've heard is that we have fucking enhancers. I don't know what they're trying to say, bro. Okay, how can the same gene be used in different tissues at different times? Alright, let's just get all this shit. Let's get all of the influence so we can move the fuck on. Yeah, bro, I'm just wasting time, bro, just learning this shit. I just gotta learn it. Whatever the fuck it's saying, just move on. There's no point in, like, You can't even get a question on this shit, bro. Alright, bro. Clip this shit, bro. Hold on, don't clip it yet. Alright, what's up, YouTube? We're gonna learn about gene regulation and development. So basically, the genes in the freaking zygote, there's things that affect it. So there's the things that are intrinsic and the things that are extrinsic. Extrinsic shit is like epigenetics and like DNA methylation and asymmetric cell division. It basically gets like uneven and lots of stuff, and you can get mutations, right? And extrinsic stuff is basically just anything extrinsic, basically outside. Environment, nutrients, and all that, and like in the matrix as well. What's there, right? So things that affect it, things that are outside of it. All right. So basically saying, you start off with a couple. Well, at the beginning, all your cells are the same, right? So how do our cells become different types of cells, right? If they're all the same exact cells and they're all the same DNA, how do they become different? Well, basically control which genes are expressed. It's only basically by what genes are expressed. That's the main driver of how the cells become different cells. And so, at first, the early embryo does not actually use its own genome. Like, so the DNA, like the genome it has, it doesn't use it at first. So, my hypothesis is that, since, like, it just became a zygote, all that chromosome is super compact and it's not able to use it. And so, it uses all the mom's mRNA and proteins, and then around the cleavage, it starts doing cleavage. And then on the stage 4 to 8, that's when it does something called like some type of like embryonic genome activation. That's why she uses the genome, right? And so then right after fertilization, this makes no sense. Methylation is blocking these. This is very confusing to me, but to remember, if you want to remember, what to actually remember from this is that basically the genome embryonic shit, basically it demethylates everything so that I can actually use it, but shit that was actually stays methylated, that's like genomic imprinting. So that moves on to a thing genomic imprinting, or if it's like on, especially like on specific on a specific dad or mom gene on their chromosome shit, if something is methylated, then it's not going to be expressed. So basically, it masks the other parent's thing. So parental, like, so then these are the terms, right? Father imprinting and mother imprinting. If it's father imprinting, then you're not going to see shit from outside, and if it's parental imprinting, you're not seeing shit from like non-imprinted, right? Parental imprinting, I can see anything from that side. Alright, so then we're moving on to the next topic. Well, some girls already have 2 X chromosomes. Well, guys only have 1 X chromosome. So, and X chromosome is a very huge gene, very huge chromosome, and it has a lot of genes. So basically, if whatever girls need to make their proteins and everything, they're gonna have double the amount of guys. I don't think it's like a guy versus girls issue. It's the fact that like they want to keep it the same amount. You don't want to have too much, right? So basically, every single cell in the body of a girl, basically, well, every single cell is gonna have chromosomes, right? It's gonna have 2 copies of chromosomes. So basically, every single cell in a girl's body goes through something called X chromosome inactivation. Basically, one randomly, it doesn't matter which one, the X chromosome of dad or mom is gonna be silenced, and you just get a normal amount of things made. Yeah, so this is a topic I was trying to like come back to. Well, every cell, every diploid cell, right? Every somatic cell has 46 chromosomes. 23 are from mom, 20 from dad, 22 are autosomes from dad, and 22 autosomes from mom, and 1 sex chromosome given from dad, and 1 sex chromosome given from mom. Right, so basically that's really it. Yeah, we can move on. Well, how can some genes be like targeted in different areas? Like you can have the same gene in different areas. How can it be the same gene used in different areas? Basically, it's all by enhancers, right? Enhancers are areas of DNA that are not coded, or non-coding DNA that basically affected by transcription factors. Basically, this image is basically just tells us that like, I don't know, right? You have an enhancer for a specific. Usually, this is the gene. It's enhancer for that, enhancer for that. So if you block this, and you're gonna get no more expression of that. If you block this, you're gonna get no more expression of that, right? Alright, so now it's not just like genetic shit, it's also like environment that affects your genes. It's very obvious, like your diet's gonna have a big effect, like what you eat and everything, the temperature you're in, environmental shit, it's all gonna affect it, right? So one main example of an environmental effect is the diary, like intake of dietary folate, right? I don't really know what it is. I think it's like some green, vegetable, vitamin type shit. Basically, the whole point of this is that they're saying is that like babies and shit, like in the mom, if she's not taking folate, then the neurotube, like around week 3, 4, when it has to form, gets fucked up. So like your baby's gonna have a lot of issues, like on the nervous system side, right? So if you just take a supplement, that's pretty what it's saying, right? Environmental has an effect on it. Alright, so basically that's what it is. So we can move on to cell signaling now. Just make that talk real quick. So we can start tomorrow. I know you're tired now. But yeah. I'm gonna just clip whatever video I made and just move on. Okay, I don't know how to end the stream now. Alright, so I gotta stop broadcasting, y'all. Alright, thanks for watching. I appreciate it.